So welcome to the next session. The next session is going to be on protein purification. So the learning outcomes of this session are to understand the applications of protein purification. The second objective is to explain the different steps involved in protein purification, and you'll discover the term CIPP. And also to explore methods to confirm the purity of a protein. I'm just going to take my laser pointer. So why purify a protein? Well, it's really important to purify protein from a mixed population. Now you may want to study its function. So obviously if you have other proteins there will be a contamination, and so we need to just have a single protein population. We may want to analyse its physical properties, so we may want to get some information about the pharmacokinetics if we're looking at kind of drugs that are targeting our protein. And also we may want to determine its sequence, so to determine which part of the sequence is important in the kind of protein structure. Protein purification is particularly useful in the biotech industry, and in particular for industrial and therapeutic applications. For medical applications, we purify proteins for drugs, and this is known as biopharmaceuticals. So things like hormones, for example, human growth hormone, antibodies which are used in, for example, in immunotherapy, and also growth factors. But there's a whole array of different types of biopharmaceuticals. It's also useful for the food processing, so things like pectinases, so purification of pectinases, as well as in textiles and leather goods. And we purify things like proteolytic and lipolytic enzymes. Also in the production of detergents, so things like amylases and cellulase. As well as in paper manufacturing and recycling, so things like silonases, and also in adhesives, so for kind of production of natural glues, as well as in kind of bioremediation, so this is treating pollution with proteins. So protein structure and purification. So the protein structure is very complex, and so to have the charge and polarity that causes them to interact with other atoms and molecules. Now you might have come across the term hydrophilic, which means water-loving, and hydrophobic, which is water-hating. Now proteins essentially are made up of these hydrophilic and hydrophobic charges, and proteins must be extracted in buffers to essentially help them to maintain their protein native structure. So how do we do this? Proteins are actually typically extracted in buffers that are at physiological pH. So normally we use a buffer of 25 millimolar phosphate at pH 7.4, which has a low salt concentration, anything between 25 and 150 millimolar sodium chloride. Now these conditions help to prevent something called protein aggregation. So we kind of discovered that in the first semester when we were looking at protein aggregation in one of the practicals. And protein aggregation is actually a kind of a bottleneck of protein purification. So protein extraction and preparation before purification. Now there's a number of steps that we have to do, but they may depend on what is our kind of starting material. So our proteins may be made intracellularly, so they may just be found within the cell. And so we need to actually look at how we can extract those proteins. Okay, so we've got lots of barriers to kind of cross. Whilst extracellular expression, this is much easier because the proteins are actually secreted into the media. So we're going to follow the ways in which we can purify these different types of proteins. So intracellular expression, we may have essentially 4 different types. So we may have some which are insoluble in the cytoplasm. We may have some which are soluble in the cytoplasm. We may have some which are membrane bound, and then some which are kind of moved to the periplasmic space in the case of prokaryotic systems. In the case of extracellular expression, the proteins are actually secreted into the culture media. Now we have to think about, well, in particularly there's more steps obviously when we look at the intracellular expression. So our proteins obviously stuck inside the cell, so we need to release the proteins. Okay, so we need to kind of break those cell walls, those cell plasma membranes, and so we need to perform something called cell lysis. Okay, we do that with insoluble, soluble, and membrane proteins. Whilst with the periplasmic space, we need to disrupt that cell wall, and then there's like another step after that. So once we've disrupted the cell wall or we've lysed the cells, we need then to remove any debris or remove any like non-disrupted cells. So once we've lysed the cells, then we need to remove the cell debris, and that's quite easy. We can do like a centrifugation. So centrifugating the sample will actually pellet any components that are not lysed. Okay, so they will be pelleted in the bottom of the tube. In the case of like extracellular expression, here we'll just take the culture media, okay, and we'll also have dead cells, which will also be floating in the culture media. So we'll also need to perform a centrifugation, and that will remove any cells. The next couple of stages, again, they will depend on what our starting sample is. So if we have insoluble protein in the cytoplasm, it's possible that we have inclusion bodies. So we need to harvest those inclusion bodies. If we have soluble proteins, then we'll just have to, after the cell debris removal, we then need to recover the supernatant. Again, this is actually after the centrifugation, so we just leave the pellet in the bottom of the tube, and we rescue the supernatant, which is the solution above the pellet. If there's like membrane, then we would have separated the sample which is membrane bound, and then we need to do like a membrane preparation. And then if it's in the case of a periplasmic space, we've done like the cell wall disruption, cell wall removal, and then we have to recover the clarified sample. And the same is also with the extracellular expression. We need to recover the clarified sample. The next step is really kind of more related to if we have insoluble, so like we had the inclusion bodies or we had the membrane preparation. So here we would need to then solubilize. Okay, so we need to solubilize the inclusion bodies and also solubilize the membranes. And then afterwards, in the case of inclusion bodies, we'll have like misfolding of the protein. So we need to refold the protein. Okay, and then if we have membrane preparation, again, we need to kind of clarify the sample. And you can see that there's different steps depending on how we've expressed the protein or how the protein is expressed in the host system. Now, the host system may be a prokaryotic system, for things like E. coli, or it may be a eukaryotic system, so mammalian cells can also be used for this function. So we're going to kind of focus now on different techniques, and in particular we're going to focus on how we can lyse our cells. So looking at cell lysis, so cell lysis is the rupture of that cell wall and the plasma membrane. And essentially we want to release the content, so the organelles and all the proteins. Okay, so we're going to release and we need to do that by different ways, and depending on the sample again. So there are a number of physical disruption methods that we can use. So we can use the pestle and mortar. So this is particularly useful for plant cells. And what that involves is we take some, say for example, a plant leaf, we dip that into liquid nitrogen, so we freeze it for a few seconds, and then we tip it out into the pestle and mortar, and then we grind it up into a powder. Once it's ground up to a powder, we need to do this quickly before it kind of defrosts, and then we need to then add our grinding buffer. There are other methods, so for example, sonication. So sonication can also lyse our samples, and we can do that with any type of sample. Essentially, it's using like sound waves, and we need to wear like special ear protection for this. And this actually, the good thing about sonication is that it will actually completely disrupt the sample. Okay, so break open those cell membranes using the sound waves, and then also it will also break the DNA as well. So it's a really useful method because we can actually chop up the DNA by using this method. Other methods are liquid homogenization, and there's different systems for liquid homogenization. Now there's two here which are using essentially a piston which is inserted into a tube. Often the piston is about the same size as the inner diameter of the tube, and the sample is placed in the bottom, and then the piston is moved up and down, and the sample is kind of the cells are kind of squeezed past the piston, and this kind of the action kind of helps to break open those cells. We also use a special buffer as well, which is a kind of like osmotic buffer, which kind of facilitates the kind of the cell lysis. And another type of liquid homogenization is the French press, which is also based on a similar system, but it uses pressure with a piston, which kind of passes, which kind of squeezes the sample, and then this is sufficient to kind of to break open those cells. And then another method which is kind of related to the liquid nitrogen is to freeze thaw at kind of very low temperatures of like -80. So we can put our samples at -80 and then take the sample out, and then we can freeze thaw, and that's depending on the type of samples that we're using. So protein purification essentially is made up of a series of processes intended to isolate a single type of protein from a complex mixture. Now, protein purification is vital for the kind of characterization of, as we saw, the function, structure, and the interaction of proteins of interest. And the starting material is usually either a biological material or cell line or microbial cultures. Now, this essentially is a three-stage process. So it involves first, we've got the preparation, extraction, clarification. We have then a capture. So we need to isolate, concentrate, and then stabilize. And then you'll have an intermediate purification. So that's to remove those kind of cell debris. So kind of remove the bulk impurities. But then we'll go on to do the first step of purification. And then the last step is that kind of the polishing, so achieve kind of a very high level of purity. You can take a look at this link here. It's a biology discussion. It's quite an interesting read, so have a look at that later. So looking at the protein purification strategy, so proteins are complex and they actually have widely different properties depending on the protein. And so no single method for preparative purification. So we need to kind of fine-tune the method to the protein. And so essentially it is a dark art. The differences in proteins can be exploited to separate a target protein from other substances in the sample. And in fact, separation usually involves chromatography, and that's based on the difference in the following properties. So, for example, the ligand recognition, so it's a specific or non-specific recognition. We can also use metal ion binding. Charge is particularly useful. Also size. So size of a protein, all proteins have different sizes. Some proteins have the same size, so maybe it's not always the best method to use. We can also look at hydrophobicity, and as well as isoelectric point. So column chromatography, essentially a protein mixture is applied to the column. So if we take the column, so for example here, we've got the column, we've got a sample which is applied to this solid matrix, Okay, and at the bottom of the column we always put like a porous plug. Okay, and we can then remove, so essentially remove this this plug, and we have a collection at the bottom. The solvent buffer is applied to the top. Okay, so when we add our sample will start to drip through the column, and then the solvent buffer is then applied to the top of the column. Okay, and then the column starts to drip, okay, into like a test tube at the bottom, and different proteins then can interact with this column at different extents and flow rates at different rates. And so the column kind of starts to separate, and then the proteins then are collected into different fractions. Okay, so we then can separate. Here we've got this. Here we've got the green protein, and then the red protein and the green protein. You can see that the red protein has been eluted off first, and then the green protein is eluted off next. So we separate them into different fractions, and that's a way of purifying or separating those protein populations. So the invention of chromatography, the invention of chromatography was in 1901 by Michael Sweat. And what he did was he separated various plant pigments. So you would have done this experiment possibly, or you know about this experiment. So separated plant pigments such as chlorophyll, xanthophylls, and carotenoids. And when we think of chromatography, the name in Greek means chroma, which means colour, and graphene means writing. Okay, so chromatography is essentially separating the components of a solution by colour. So the original chromatography, we started off with a glass column filled with powdered limescale, so calcium carbonate. Ethanol extract of the leaf pigments is applied to the top of the column, and the ethanol is used to elute the pigments down the column. Okay, so what happens? So after some while, the elution, at the end, we get essentially a series of coloured bands which are visible, which correspond to different pigments in the plant extract. Okay, so we get the different coloured, and the bands were found to be chlorophylls, xanthophylls, and carotenoids. So some of the common chromographic methods, so some of the very simple methods, we can use things like paper chromatography to separate our products, thin layer chromatography, TLC, you might have used this before in the past. We can use high performance liquid chromatography. Okay, so that's using, it's a kind of fast TLC. We can use gas, so liquid chromatography, GLC, or fast protein liquid chromatography, known as FPLC. So chromatography involves essentially a solid stationary phase and also a liquid or gas mobile phase, so known as the Touette's plant pigments. There are different solids which can travel different distances through this solid phase, and each solid has an equilibrium between the absorption on the solid phase, so on the solid surface, and solubility in the solvent. And this is represented as an equation. So we have S, which is the mobile phase, which is in equilibrium with the stationary phase. And we can make a calculation of the partition coefficient. So we can say, okay, well, the stationary phase divided by the mobile phase. And when we look at a chromatogram, we will observe different peaks. The first peak that we'll observe is called the mobile phase, so the TM, and it's we have normally a detector signal on the y-axis, and on the x-axis we would normally have time. And TM is the time taken for mobile phase to travel through the column, so for the mobile phase to travel through the column. whilst the retention time, which is TR, okay, this is the time taken for the solute to elute from the column, okay. So we have two things going on here. So we have the time, the mobile phase, and the retention time. Looking at asymmetric peaks, so solute elutes as a symmetrical Gaussian peak, and these are the ideal conditions. However, we can get other types of peaks, and this happens when the solute partition coefficient, KD, is the same for all solute concentrations. If this is not the case, then the peak has an asymmetric shape. So we can have something called peak tailing. This is where the stationary phase is not homogeneous. So you can see here, this is an example of a peak tailing. Or we can have a peak fronting. So here, this is where the stationary phase is overloaded. Okay, so this is where you can see the peak fronting here. Looking at the retention, so essentially the retention factor is a capacity factor known as K prime, and it's a peak retention measure. So the larger the K prime, the longer the retention time. Typical column material are things like silica or alumina. And K prime is kind of represented as this equation. So it's the retention phase minus the mobile phase over the mobile phase. Now, if we have a K prime of 1 to 5, that is ideal. However, if we have a K prime of 20, then it's very slow. Or if you have a K prime of less than 1, then it's too fast. So we need to have something around between 1 to 5. So looking at the resolution, so chromatography resolution. So resolution is known as RAB, and that's between two chromatographic peaks A and B, which is a measure of how well A and B have separated. So we can see here, these are the kind of detected responses and the peaks. So here, these two peaks have not separated well, and you have an R value of 0.5. Whilst if have an R value of 1, again, there is an overlap between the two peaks, so there is possibility there is not very good separation. Whilst with an R value of 1.5, we can see that there is good separation. And we can measure this using this equation. So RAB equals TRB minus TRA divided by 0.5 WB times WA. Which then, if we solve that equation, it would then give us delta TR divided by 0.5 WB plus WA. Now TRB in this equation is the later eluting solute, whilst W is the peak width. So to improve resolution by increasing delta TR or decreasing WA and WB. So A is like the original. So if we decrease WA and WB by 4 times, so reducing the kind of the peak width, we see something like this. But if we increase delta TR by 2 times, then we'll see something like this. So this is something that's kind of ideal. Focusing now on different types of chromatography. So we're going to visit different types of chromatography. And the first one is ion exchange chromatography. So ion exchange has a coated solid stationary phase. So essentially this is the resin, okay? And the ions, which can be cations or anions, they're bound to the resin. So this is like CO2 minus, okay? And the ions with opposite charge bind to the resin surface with Na sodium ion plus. And the elution of the mobile phase, which is always liquid in the ion exchange, is through this resin. And what happens, it displaces the sodium ions by other ions. Okay, so M is just like another ion. So the sodium ions get displaced off the ion exchange column. Molecular exclusion, so this essentially is a mixture which passes as a kind of gas or liquid through the porous gel. The pore size allows large solute particles to pass through uninhibited, whereas the small molecules are slowed down. Okay, so the separation is according to particle size. And so here we have the mixture of particles as the direction of flow through the column. So this would be made up of our gel particles. And you can see now as the separation starts to, as the solute starts to go through the column, we can see a separation of these particles. So you can see that the molecules which are larger than the largest pores of the swollen gel particles, whilst the molecules which are small enough, they can penetrate into the to the gel particles. So usually there's several steps that are required to completely separate a target protein from all other substances in the sample. So these are kind of proteins to be purified. So this is a target protein that has a known ligand. So when we have a known ligand, then the initial approach is to use ligand recognition. If the target protein is a recombinant and contains a tag, this is quite good. We can use different types of tags. So we can use things like metal ion binding in the case of histidine tag, or ligand recognition depending on the tag. If we have a target protein that has no tag or no ligands, then we need to then use other types of purification, and this is where size exclusion and charge come into play, and they can be applied in a suitable order. Focusing now on affinity chromatography. So affinity chromatography essentially is the reversal interaction between a target protein and a specific ligand attached to the chromatography matrix. This increases concentration of target proteins. It is biospecific, so it relies on an antibody binding to an antigen or a lectin binding to a particular sugar group. If it's non-biospecific, protein binding to a dye substance, you could have histidine containing proteins which bind to metal ions, and we'll revisit this later. So this offers like high selectivity and also high resolution. And in fact, this is often used as a first purification step, which is then followed by a polishing step, which then removes any remaining impurity and aggregates. So let's look at how affinity chromatography works. So here we've got the flow of the mobile phase. It's the top of the column, this is the bottom of the column. Okay, here we have our beads which have an antigen or an antibody bound to the beads, and we have got A, B and C proteins. Okay, so A will start to move through the column and it doesn't get trapped. B will move through the column, while C will move through the column and will get trapped within the column. Okay, so there is these ligands will be recognized, so that if it's an antibody, there will be an antigen interaction. So when we look at a typical chromatograph, so we can use absorbance to measure the elution of proteins off the column. And often we use this on the y-axis, and then on the x-axis we have column volumes known as CV. So what we do is we start off with to elute with 2 column volumes. And this is where we kind of begin to add the sample. Afterwards, we get we need to get equilibration. So you have absorption of the sample, and then you have like the flow through of the unbound material. And then afterwards, we get washing to remove any unbound material. So essentially what we'll see is we'll see the first peak, essentially this is the material which is coming off the column, okay? And then afterwards, this then should flatline. And this will take several column washes, so between 2 to 5 column washes. Afterwards, then we need to change the buffer to a elution buffer, and depending on what is bound, we can use a specific elution buffer. So this is eluting the bound proteins, and essentially we need to elute them in a kind of very short peak, normally in less than 1 column volume. And then there's like a regeneration of the media, so then we can wash the column. Go back. Oops, sorry. We can then wash the column with 2 to 3 column volumes. So now looking at affinity chromatography tags. Now there are different types of tags that we can use in the lab, and some of the common ones are included on this slide. The first one is the polyhistidine. So this is made up of 6 histidines as part of a tag, which is then fused to our protein. Now the 6 polyhist tag combine metal ions, so to nickel chelated support, so we can use something called a nickel column to purify our protein. GST or glutathione S transferase, this also is a tag, and this binds to glutathione, which is used as our support on the column. We can use immunoglobulins, so the FC portion of immunoglobulins can bind to immobilize protein, so things like protein A or protein G. So protein A may be used as a fusion protein because this binds to the immobilized IgG FC region. Also biotin. So biotin can be used, is quite a popular support. It binds to streptavidin. Streptavidin is a bit like is an antibody which we can be bound to the column. We can also use sugar units. So we can use lectins to purify the sugars. Looking now at the affinity chromatography using GST tag, the glutathione tag. GST tag proteins bind to glutathione on the column. So here we've got our porous bead with the glutathione and the GST tag on our protein. You also have something called a thrombin as well, so I'll come to that in a moment. Non-specifically, or it kind of binds non-specifically, or weakly bound proteins will be washed off. So the protein will be bound to the column, and then the other proteins will be washed off. Next, we need to elute. So to elute the bound GST tag proteins, we can use either glutathione as a competitor, so to compete off the bound protein, or we can use a protease known as thrombin. So here I told you there was a thrombin site, and so thrombin is a protease which can actually cleave this, and then will cleave off our protein, which then can be eluted into various fractions. We can also use something called immobilized metal ion affinity chromatography, known as IMAC. Proteins containing histidine residues, they combine to immobilized divalent metal ions, so such as nickel, copper, zinc, or cobalt. The hist tag proteins, they have there's multiples, so you have anything between 6, which is common, to 10 histidine residues, which are usually the stronger binders in a crude protein extract. And this is what we can get these manufactured columns from pharmacy, which are useful for purification. Essentially, they're filled with Cephros beads. The Cephros beads have then linked to have immobilized metal ions, and the metal ions, so here this is nickel, they can then pick up the histidine tags, so the histidine tagged protein. Kind of looking at a chromatogram of like iMac creating purification. So when here we're monitoring the absorbance of our column. So these are the things which are being eluted off our column. So remember, first we kind of have this kind of stabilization. So where we've kind of eluted proteins which are not bound to the column, and then we wash off, wash the column. And then what we do is then we add something called imidazole. So imidazole helps to essentially elute our protein. And we first have an initial peak here. It's not really a peak, it's just a kind of a little kind of hill. And then afterwards we then kind of elute our target protein with imidazole. So by increasing concentration of imidazole, this is then used to compete off the His tag protein. This is competed off, which is bound to the nickel column. Here's the non-metal binding proteins. So these are the proteins which don't have the histidine tag. And here, this is the His containing metal binding proteins. Okay, so you do get some which do have histidine, which will bind kind of like non-specifically, but they will kind of be competed off. And then this is the 6-histidine, which contains the target protein. Focusing now on size exclusion chromatography. This is also known as gel filtration. It can be used to separate substances with different molecular size, and they can be fractionated into different by different particular size range. And this is to remove all the molecules larger than a particular size from a sample or combination. It's a powerful step because we can use this after affinity chromatography to polish the sample away from any impurities. And rarely we use this as a first purification step. There are different columns which are available and they have different sample volumes. We have non-binding method, which is there's no concentration of the sample takes place, and then afterwards we have a dilution of the sample results. The load sample volume must be kept small, so that's really important, anything between 0.5 to 2% of the total column volume. The buffer has low salt concentration, so between 25 and 150 millimolar. And remember that was useful for kind of keeping protein structure. And this also prevents any weak electrostatic effects between the protein and the column matrix. So now running through a gel filtration chromatography. Here we have a concentrated sample, which is a mixture of A, B and C protein. Here we have the stationary phase. This is the flow of the mobile phase. The sample contains 3 components, A, B and C, which are then placed in the top of the stationary phase. As the sample is carried through along the column of the stationary phase, the components are spread out, okay, according to size. And at the end of the column, the components are then separated. So this is what we would see on the chromatogram as they come off. If we look at the flow of the mobile phase, so here we take large molecular weight proteins which kind of pass through the cephase. Now the large weight will pass through and exit quickly with the mobile phase, so normally in less than 1 column volume. Intermediate molecular weight proteins, they pass through the cephase beads, and occasionally they enter the beads of the interior and they're kind of slowed down the elution from the column, so less than 1 column volume. Whilst the low molecular weight proteins, they pass around the cephase beads and in and out of the bead interior, and they vastly slows down the elution from the column and that's greater than 1 column volume. So you can see how we can separate the different sizes according to this gel filtration chromatography. If we take a look at a chromatogram, what we see initially, so here we've got the absorbance and then the column volumes measured as CV. First we have an equilibration of the column, and then afterwards then we add our sample. So a sample is injected into the column, and then here we have the first peak. So this is the high molecular weight proteins. Afterwards we have some, we when wash with 1 column volume, we have some intermediate molecular weight, and then afterwards then we have our low molecular weight, which then are eluted last. Kind of focusing now on ion exchange. So ion exchange chromatography separates proteins with differences in surface charge. So this is kind of a high resolution. So ion chromatography helps to retain analyte molecules on the column based on something called ionic interaction. So the interaction with a charged protein and oppositely charged resin. So target proteins are kind of like concentrated. Isoelectric point, the net surface charges vary with the surrounding pH. So remember the pH scale of 1 to 14. Okay, a protein at pH 7 has a net charge of -1, whilst at 1 it has a net charge of +1, whilst at 14 it has a net charge of -6. Ones in between, we have net charge, so between 1 and 7 has a net charge of 0, and this is known as the isoelectric point. So it's useful for anion exchange because it will bind to positively charged column matrix. So it's useful to know some information about our protein, so in particularly the isoelectric point. So if we look at how the protein kind of moves through this column, here we've got our kind of non-charged particles. Our positively charged particles will move through the column, whilst our negatively charged proteins will actually be retained on the column. So the principles of ion exchange chromatography. So above its isoelectric point, a protein will be negatively charged and will bind positively charged to an anion exchanger. If it's below its isoelectric point, a protein will be positively charged and will bind to a negatively charged cation exchanger. So proteins are loaded onto a column at a low ionic strength, so low salt, and the elution is then performed by a gradient of increasing salt concentration. So if we look at this chromatogram, so this is something that we saw before. So here we've got we've actually washed the column with 10 to 20 CV volumes. So we have an equilibration step, and then we add our sample to the column, and the unbound molecules are eluted before the gradient begins. So as the gradient begins, the tightly bound molecules are eluted in a high salt wash. Okay, so the tightly bound ones are only released in the high salt wash. And then afterwards you have a re-equilibration, at which point you can then reuse the column, and that's normally at least 5 column volumes. Hydrophobic interaction chromatography. So what is this? It separates molecules based on the hydrophobicity, and each protein has a different hydrophobicity, and it's enhanced by ionic strength buffer. So typically we use ammonium sulfate or sodium chloride, and these are normally concentrated buffers, so either 2 molar, 1 to 2 molar, or 3 molar sodium chloride. It can be used as a first step in intermediate step or as a final step, so resulting in the target protein, which is then concentrated. So here you've got you can see the hydrophobic surfaces on the column, and we have our kind of highly ordered water and the less ordered water. And then when we have high salt, what happens is in the high salt it gets bound to the column, and in low salt it's soluted. So the final result of Higgs separation is based on the interplay between the distribution of the surface exposed hydrophobic amino acids, the hydrophobicity of the medium, and the nature and composition of the sample, as well as the type and concentration of salt used in the buffers. The elution is usually performed by decreasing the salt gradient or adding something called chenotrophs, such as urea, or we can use guanidine hydrochloride. Now, these help to denature proteins, so by reducing the level of interaction. So if we look at a chromatogram, so this is showing first we have the equilibration, so that's with 5 column volume washers. Afterwards, then we apply our sample, and next then we have the unbound molecules which elute before the gradient starts. Then we have the gradient elution. And then afterwards we have like a salt-free wash. And the tightly bound molecules only elute in the salt-free conditions. Reverse phase chromatography, also known as RPC. It's also known as hydrophobic chromatography. It includes a kind of chromatograph method that uses hydrophobic stationary phase. The separation is on the basis of hydrophobicity. It's quite similar to HIC. However, reverse phase media are much more hydrophobic. It's a high resolution method, and it requires the use of organic solvents. It's widely used to check for purity when the activity and tertiary structure are not a focus. Proteins tend to denature, and they can actually bind quite strongly to the reverse phase chromatography medium, and they can be actually difficult to elute. For small proteins, they are less commonly denatured by organic solvents, so actually reverse phase is actually an excellent choice for kind of the final polishing step. So looking at how this works, so here we have our matrix and then the hydrophobic surface. Normally we have our sample up to 5% acetonitrile. As we increase the organic modifier content, what happens is we get more hydrophobic sample and also less hydrophobic sample. And the initial conditions are kind of highly aqueous, so 100% will bind to all the hydrophobic substances in the chromatography matrix. But by increasing the solvent concentration, we increase the hydrophobicity of the mobile phase, and so the proteins then interact less with the matrix. So it's a good way to kind of as a kind of polishing step to kind of remove any remaining contaminants in our sample. So eventually then the proteins then can start to elute, with the most hydrophobic proteins eluted last. If we take a look at a chromatogram of the reverse chromatography, again, here we have the 5 column wash to kind of equilibrate the column. Afterwards, we add the sample to the column, and first we have a kind of increase in gradient dilution. Okay, and the least hydrophobic proteins are then eluted first, whilst the most hydrophobic proteins are eluted last. Okay, and then remember we have then 2 column washers to equilibrate the column. Okay, so that kind of concludes the purification, but we need to kind of consider how do we confirm the purity of a protein. And the chromatogram shows us the absorbance of a protein products, but that they're eluted from the column, but which ones? We need to kind of verify what are the proteins that we're looking at. And so we use SDS page to check for the purity of the protein. So we can check to see if there's any other proteins in our population. So what is SDS? Well, SDS stands for sodium dosyl sulfate polyacrylamide gel electrophoresis. It's also often written as SDS page. It's a type of electrophoresis, so this is a laboratory technique for separating molecules, and this is based on the charge. So an SDS apparatus, you've probably come across this before, essentially consists of 2 tanks. So here we've got the cathode and we've got the anode down here. Now the proteins always migrate from the cathode to the anode electrode. What we do is we create a chamber at the top here, and here we load on a gel. So we pour the gel, and it's normally made up of polyacrylamide. We then pour our gel, and then we load our samples into a well on the gel. What happens is when our samples are loaded, well, first we need to do something to our samples. So if we take our samples, we have a mixed population A and B and C. We may have disulfide bonds between A and B, or even within the protein itself. Then we need to denature the protein. So we need to separate these proteins and then also kind of essentially denature them, so break those disulfide bonds. And to do that, we heat with SDS and something that will break the disulfide bonds, such as beta-mercaptoethanol or DTT. What the SDS does, it actually coats the protein in a negative charge, so it coats it in SDS, which then allows it then to kind of separate according to size as opposed to charge. And the beta-mercaptoethanol breaks those disulfide bonds. And so essentially then we can then separate the proteins according to size. And afterwards we can then take the gel from the tank, so once it's run through the gel and the different is run off, then we can take the gel and then put it in a special staining solution, and we can use something like Kamasi blue. So Kamasi blue is for kind of proteins around about 200 nanograms, while silver staining is for kind of lowly expressed proteins. So with the Kamasi blue, essentially we incubate the gel with the Kamasi blue for a couple of hours, and then we use a destain, which is made up of methanol and acetic acid. We destain the gel, and then afterwards we can then take a picture of our gel. And here you can see samples which have been loaded. They don't actually indicate on this gel the molecular weight, but here it's actually been indicated. So normally we'd observe a lane with the molecular weight marker. Here you've got the larger proteins, which are 100 kilodaltons or 100,000 daltons. Here's one at 40,000, one at 15,000. And if we look at these are samples which have been run on a column, and the different fractions have been like kind of separated, we can see that here in this first fraction, this is kind of like the unpurified protein. This would be the first step of purification, second step, and you can see that there's a major product of about 40,000 daltons or 40 kilodaltons. And you can see that for each step there is a purification, and this is removing all these contaminating proteins from our sample. Kind of based on understanding how the proteins are kind of charged. So the separation of charged molecules. So the charged molecules are separated based on the electrical charge and size within the matrix. So we have a mixture of charged molecules. We can separate them according to charge. We have positive molecules and negative molecules, which then we can analyze, identify, and purify. Whilst the gel matrix is composed of either agarose or polyacrylamide, but mostly we use polyacrylamide. For the agarose, we can use agarose for separation of proteins, whilst the polyacrylamide has smaller pores than agarose, and therefore you have a higher degree of resolving power, so meaning you get better resolution. So this is a comparison of agarose and polyacrylamide. So agarose has been extracted from seaweed, so it's a polysaccharide. It has to be casted horizontally. It's non-toxic. It's ideal for separating large molecules, so if you have large molecular weight proteins. It is actually commonly used for DNA separation, and staining can be done before or when you pour the gel. Whilst polyacrylamide gels, this is a cross-linked polymer of acrylamide. The gel is cast vertically. It's a potent neurotoxic. It separates small molecules and is used for DNA or protein separation. And the staining can be done after pouring the gel. So for protein electrophoresis, separation is based on size, so this is molecular weight, and it allows us to characterize, quantify, and determine the purity of the sample, as well as comparing proteins from different sources. So it has multiple uses in protein purification. Proteins, unlike DNA, do not have a constant size to charge ratio. So in an electric field, some will move to a positive and some will move to the negative pole, and some will not move because they are neutral. So the native proteins may be put into gel systems, electrophoreous, whilst an alternative to native protein gels, this forces all proteins to acquire the same size to charge ratio. So SDS page, the purpose of this method is then to separate proteins according to their size and no other physical feature. So the SDS, which is the detergent, this has a negative charge, the sulfate, which is attached. And if the SDS is added to the proteins, they will be solubilized, plus all the proteins will be covered with negative charge. So as I was telling you before, we denature our sample, we also add the SDS, and then our sample is then coated in the negative charge. So the SDS coats our protein in this negative charge, and so if we run our sample, the sample will run from the negative to the positive electrodes. When we look at this is an example of an SDS page gel. So this is a separation of different proteins. So the proteins at the top of the gel are the larger proteins. The ones at the bottom of the gel are the smaller proteins. Okay, so here we've got loaded 1, 2, 3, 4, 5, 6. Here we've got catalase, which is cytochrome C. So here we've got catalase, cytochrome C. And alpha-lactalbumin. And what we can do is we can actually identify different species within these lanes. So, for example, we've got catalase, we've got cytochrome C. Also in lane 2, we've got cytochrome C and alpha-lactalbumin. So we can look at which bands are common between these samples. So you can see here this band, and this band, this band, and this band are quite common between those samples. In the next lane, we've got BSA, cytochrome C, and alpha-lactalbumin. Okay, so we can identify that probably, what do we reckon, alpha-lactalbumin. So these proteins here, they might be these proteins here. But, it's difficult, obviously to know what those proteins are, but we can actually look to compare the sizes of these proteins. So in summary, proteins can be separated and purified on the basis of the charge. So we can use ion exchange, known as IX. We can use specific ligand recognition, so this can be bispecific or non-bispecific. So this is things like affinity chromatography. We can separate them according to size using gel filtration or gel exclusion, size exclusion, sorry. We can separate them according to hydrophobicity, so we can use HIC, hydrophobic interaction, or reverse phase, RPC. We can also separate them according to isoelectric point, so this is known as chromatin focusing. Okay, so this is coming towards the end of the session now, and this is some further reading, suggested reading. So please have a look at Berg et al. It's a 2015 biochemistry, so exploring proteins and proteomes. So have a look at chapter 3, pages 65 to 76. This book's available from the library. I've uploaded this handbook from GE Healthcare, and it looks at strategies of protein purification. So have a look at this handbook. It's quite useful, and further reading around different types of protein purification. Also, there are some additional information on the blackboard, so have a look for this week. Okay, so thank you for listening.